last time we started to discuss lipids, which lipids, remember, are fats. And did we talk about fatty acids at all, or did we not? I can't quite remember where we were at. We didn't get to talk about them? Yeah. We did. I thought so. Okay. So, fatty acids are important, even though they are the simplest, because we utilize them to make the next two types of fats. Okay. Something that's different with fats than you see in the other organic compounds is that there isn't a standard monomer to build all fats. Okay. These are as close as it gets. Okay. Remember, a monomer would be like a brick building up to a brick house. In carbohydrates, we had monosaccharides that built disaccharides that built polysaccharides. And we don't see that for all fats, but these are as close as we get to those initial building blocks. And they do build triglycerides, which a triglyceride is made up of fatty acids, 3 of them, hence the name triglycerides, plus a 3-carbon glycerol molecule, which is the backbone of a triglyceride. If we look at the figure here on this page, that is what a triglyceride looks like. It includes a 3-carbon, 1, 2, 3-carbon glycerol bonded to 3 different fatty acid tails. Sometimes we call them tails. And these are the body's most highly concentrated form of chemical energy. This is the primary form of fat that we have in our body and the primary form of fat that we ingest. Okay. Triglycerides are either found as a liquid or a solid when you find them at room temperature. A fat is a triglyceride that's solid at room temperature, and that's because those fatty acids are saturated, which if we go back one slide, remember saturated, there are no double bonds. Okay, examples of fats, and these are used in cooking, like lard, okay, Crisco. Is that stuff healthy for you? No, really unhealthy for you. Okay. Oils like canola oil and safflower oil, peanut oil, okay, are better for you, and they're liquid, hence the name oils. We all know oil is liquid. That's because they have unsaturated fatty acids. Okay. Remember the shape here with an unsaturated fatty acid is with this little bend in it? That actually prevents them from solidifying at room temperature. So if you ever wonder why, now you know. Fats and triglycerides, specifically triglycerides, are very plentiful in the amount of energy you can get from them. They provide twice as much energy per gram as do carbs and proteins. Carbs and proteins only give you 4 calories per gram. Triglycerides give you 9. Okay. A calorie is basically just a measure of how much energy you can get from something. Okay. We have an unlimited capacity to store triglycerides, which is very dangerous if you don't have a good lifestyle. Because if we ingest extra carbs and proteins and fats, oils, we're going to store them in triglyceride form in our adipose or fat tissue. So we can't convert stuff to triglycerides. And it's basically a means to protect us in case we won't get food, okay, which was great long time ago when you had to hunt and find every scrap of food. But now here in modern day where food is very plentiful, it causes people to put on weight, especially if you are not burning off that energy. Similar to triglycerides are phospholipids. Similar in that they both have a glycerol backbone and that they are made up with fatty acid tails. There is a really big difference that exists, though, with these guys. Okay. Here in our figure we can see, if I'm showing you right where my cursor is, 3 carbons. There's our glycerol. There are 2 fatty acids, but triglycerides had how many? 3. The third carbon here doesn't attach to a fatty acid. Instead, it attaches to a phosphate group, which is then attached to some other stuff, which creates a distinctive head, as we call it, which is polar. We said that most fats are nonpolar. Well, now we see a difference. The tails that are fatty acids are still nonpolar. Okay? This is important because this is going to affect their relationship to water. We said that fats are hydrophobic, right? What did hydrophobic mean? Fear of water. The tails, just like other fats, are afraid of water. They don't like it. They will be seeking out places that are not watery. The heads actually love it. We describe them as hydrophilic. They have these 2 distinctive regions, which is why we describe phospholipids as being amphipathic. They have 2 distinctive polar and nonpolar parts. Amphi means both, like amphibious. Okay, so pathic in this case, I imagine, means charge. Okay, 2 different charges. Why this is important to us is going to play a big role in what we talk about next week when we talk about cells. These guys form the primary structures of the plasma membrane into what we call a lipid bilayer. Or you guys know what lipid means. What does bilayer mean then?
2 layers.
2. Okay, and it is like we see here that I drew on the board, or what you can see here on the very far right of the page, where you've got 2 layers of phospholipids with the heads pointing in opposite directions and the tails pointing towards one another. The tails, which don't like water, are going to point towards each other because the heads point where there's water. Guess what? Outside the cell, there's water. Guess what's inside the cell as well? Water. So that's going to be important for that. This is important because this is going to affect what can come in and out of the cell and how stuff can come in and out of the cell. Something that's very, very important to cellular function, which we'll get to next week. Our next type of fats look nothing like the first 3 that we've seen. These kind of look more like carbohydrates because they're kind of ring structures. Steroids, derivatives of a 4-ringed hydrocarbon skeleton. If you look here at our 4 different types, picture A, B, C, D rings, picture B, A, B, C, D rings, so on and so forth. 4 different rings. And then you've got different things attached at various points, which is why they're and how they're different. Okay. These are structurally different from the other lipids. Most common steroid in us is cholesterol. Okay. Hopefully not floating in your blood, clogging up your arteries. Cholesterol is found in places like your plasma membrane, actually. So it helps to form that lipid bilayer. It just doesn't do it quite as fancily as the phospholipids. We'll talk about that next week. Cholesterol is also important for forming the other steroids, like the sex hormones, estrogen, testosterone. Cortisol is not a sex hormone, but it's a steroid hormone that regulates various aspects and helps us combat stress. And vitamin D we get from the sun, we activate, and it's a form of steroid. Bile salts, these are actually fats that help digest other fats. That's kind of cool, and we'll talk about bile later in the semester. Don't talk a lot about steroids, though, for now. Other lipids include eicosanoids, a fun word to say. Lipids that are composed of a 20-carbon unsaturated fatty acid, known as arachidonic acid. 2 principal subclasses include prostaglandins, leukotrienes. We don't really talk too much about these guys in this class. Prostaglandins we will talk a little bit about. because of their role in regulating effects of several hormones. Leukotrienes, we don't really talk much about those, and their function is allergic and inflammatory responses. There are other lipids that include fat-soluble vitamins. We learned last time what water-soluble meant. What did water-soluble mean? Dissolves in water, so what do you think fat-soluble means? Dissolves in fat, which is something that we'll talk about next week, why that's important with regards to cells. It includes A, D, E, and K. Those are the 4 fat-soluble vitamins. Vitamin A is converted from beta-carotene. We get in egg yolks and carrots and tomatoes. Okay, we don't really talk too much about vitamins in this class, but if you take nutrition, you definitely will. Lipoproteins, I don't talk a lot about those either, but those are another form of fat. Lipoproteins are a hybrid between fats and proteins. Our body carries our fats a lot of times in our blood in this form because the proteins basically surround the fat on the inside, help it to be carried in our blood because our blood's full of water. As we know, fats don't like water, so this helps that occur. Anybody ever heard of HDLs and LDLs before? Okay, if you are involved in a nutrition class, you will definitely learn about one of these. The L for those words is short for lipoproteins. High-density lipoproteins, what HDLs stand for. LDLs are low-density. And these are measurable things that a lot of blood tests will read out. What you want are higher-level HDLs. They actually lower your blood cholesterol, which is good. You don't want high LDLs. Unfortunately, being poor in health as a result of poor diet and poor exercise does what you don't want to have the low HDL and high LDLs. So too many Americans suffer from that issue. All right, table 2.7, just a table from your textbook, just goes through and lists off all those various lipids, talks about them in greater detail. You can look through it if you want to. Third class of organic compounds are the proteins. Large molecules making up 12 to 18% of a normal lean adult body. And very important sentence right there is that they are instrumental in almost everything cells do. Almost everything cells do, proteins help with. That is really illustrated with the very last sentence of the page. Chances are, if something is getting done in your body, being performed by a protein. Now, not always, but there's a good chance that it's a protein doing it. We'll look at the next page and see what they do and examples of how they do it, who does it. Okay. Proteins are made up of amino acids put together in a linear polymorph shape. Which means they're like beaded necklaces. Okay, each bead being a amino acid bonded together in what we call peptide bonds. They're more complex in structure than carbs or lipids. And there are 20 different amino acids, and they all are going to have a similar structure. As you can see right here, you've got a central carbon. On one side is an amino group. That's a functional group we talked about the other day. Carboxyl is another functional group. A hydrogen atom. The R group, R chain, side chain, whatever you want to call it, is where variability between those different 20 amino acids comes from. This is not R for a atom. Okay. This is going to be a group of atoms, and it will vary from amino acid to amino acid. Now, you take 2 amino acids, bond them together, you got a dipeptide. 3 is a tripeptide. 4 to 9 is just known as a peptide. 10 plus is a polypeptide. Okay. They aren't technically proteins until they've given themselves specific shapes. Okay, which shape is something I'm going to talk a lot about on the next page. How many different proteins are there? Well, there's a lot. Think of amino acids like the letters of the English alphabet. How many letters are in the English alphabet? 26. Okay, so you got 20 amino acids, 26 letters, so not quite as many. How many different words are there in the English language? You don't have an answer for that. I don't have an answer for that. Okay. Because you can make words so many different ways. Okay, so many different words because all it takes is different combinations and order of letters, right? We don't know how to make words. Write them down. You guys do it every day. Proteins are essentially the same way. And we put them together specifically with the way that the amino acids are ordered in a number to give off and make the specific type of protein. Why that's important is because each protein has a unique function. And there's a great variety of proteins possible. Not only in us, but in other living things. So let's look at those functions. We've got 6 functions showing you the type of protein, what they do, and explaining what that means. You will need to know this table. I guarantee it. I won't worry about the examples, but I do want you to know the type and the function. Okay. So the first type of protein are structural proteins. A function being they form structural framework of various parts of the body. Okay. Our bones, for example, are loaded up with a protein called collagen. Your skin actually has collagen in it. Okay. Keratin is another protein. This is stuff I'm actually going to talk about this afternoon in anatomy. So, illustrating stuff that I'll talk about in here and in that class. Regulatory. A lot of hormones are proteins. Okay, I said some of the steroids are hormones. We've also got proteins that are hormones. Okay. Insulin. You've probably all heard of insulin before. Insulin is a protein. Helps us regulate blood sugar. Okay. Other ways that proteins can help regulate would be some of the neurotransmitters that our body utilizes. Neurotransmitters are chemical messengers that our nervous system uses. Okay, we'll talk about neurotransmitters in our next unit. Contractile. Okay, our muscles are loaded up with various proteins that help us move, to contract our muscles. Okay, the 2 primary ones that we talk about for that are myosin and actin. Okay. Immunological. They aid responses, protecting the body against foreign substances and invading pathogens. Your antibodies that your body produces in response to exposure to something are proteins. Transportation, like hemoglobin, which carries our oxygen, carbon dioxide. Transport means they move stuff around. Okay. And then catalytic means that they are going to speed up chemical reactions, and this includes any of the enzymes in our body, which they have shown there as like salivary amylase, which we'll talk about, is an enzyme in our saliva that helps us digest carbohydrates. Sucrase is an enzyme that we'll actually see today, helps digest and break down sucrose, table sugar, and then ATPase. There are several different types of those. We'll talk more about enzymes here in just a little bit. I mentioned that when you build these chains of amino acids, they don't begin functioning as a protein until the protein forms its specific structure. Structure is something that's absolutely important for function of a protein. And that's the case for really everything in our body. Structure affects function. Proteins are no exception to that rule. Protein's shape is sensitive to its surrounding environment. Specifically 2 things: the temperature and the pH in which it is found. Okay. Now temperature is going to affect all enzymes the same. Okay, because we just have one temperature, body temperature, that's going to affect all of our enzymes. We know that pH, though, will vary from substance to substance in our body. Like we talked about gastric juice having a real low pH. We also talked about how, like your saliva, for example, has a really different pH Okay, we'll talk about that a little bit more in a second. How they affect the shape and function is illustrated with my analogy of a slinky. Okay. Everybody probably played with a slinky at some point in their life. I use slinky for kind of two reasons. One here, what looks like a slinky is shown. Alpha helix, form of protein. Okay. Slinkies are fun when they are correctly put together. And in their correct shape, they can go from stair to stair to stair to stair, all the way down to the bottom of the steps. But eventually, slinkies are going to get tangled up or stretched out, and what happens to their ability to go down the stairs? It's not going to happen, not correctly anyway. Proteins are the same way. If they lose shape, they lose function. And that is what denaturation means. When a protein is denatured, it loses its shape, therefore loses its function. And we can see that with the image down there at the bottom, where we go from a nice coiled up protein to stretched out. Here, the top of our image, we're showing off 4 different classes or ways that we can see proteins in structure. The simplest all the way to the most complex. You can see they have these very cool shapes, and that's, like I said, important for function. Their shapes are a big result of hydrogen bonding. And the reason why we see these coils and these beta sheets, and even crazier as we get to the quaternary structures. So hydrogen bonding is not just important for holding water together, it's important for these things too. So just a second ago, I want to talk a little bit more about enzymes. Okay, one of those 6 different types of proteins. Enzymes are essential proteins in our body. We describe as biological catalysts. The catalyst is described as 3 things. The chemical that increases the rate of reaction, that's the whole purpose of it, is to increase chemical reactions. Okay. It's not changed at the end of the reaction and doesn't change the nature of the reaction or the final result. What that means is no matter if there is an enzyme or not, we're still going to go through the chemical reaction. But with the chemical reaction, it speeds up how fast we get there. I'll show you why here in just a second. Okay. And at the end, the enzyme is just like it was before. It's not changed. Okay. Now, how enzymes speed up chemical reactions is they alter this thing called activation energy, or energy of activation, which is the energy that's required for a reaction to occur or proceed. Because for every chemical reaction occurring, there has to be sufficient energy. You can think of activation energy like an obstacle. And I can show you what I mean here on the next page. Okay. Here on the left, we're showing off a chemical reaction kind of in a graph form. Remember, reactants are the starting materials, products are the ending materials. To get from here to here, we have to go over an obstacle, the activation energy. Okay. If we want to use a real-world example, I like this one a lot because it's an obstacle. I will use the Warrior Dash, or I think they do the Scotty Dog Dash. I don't know if they still do it. Different obstacle races, 5Ks, half marathons, whatever you want to call them, where you run from a starting point to a finish line, and to get there you have to complete these various obstacles. Okay. The last obstacle of our race is going to be a climb over a wall, which then on the other side of the wall is the finish line. If an enzyme is not present, it's like the wall's 10 foot tall. If an enzyme is present, the wall goes from 10 foot tall now down to 5 foot tall. Now, which of those is going to allow you to get over the wall and across the finish line faster? The 5 foot wall. That's obvious. So without enzymes, we get there slower. We still get there, but with enzymes we get there quicker. And that's because they lower the activation energy. If it helps you to highlight or underline that, that's really how they work. They lower activation energy. Now, structurally, enzymes consist of two parts, protein portion called apoenzyme, non-protein portion called cofactor. I don't really get into that too much, but it is something I do want to say, and I think it's kind of important to understand, is that we've talked about carbs and fats, and now we've talked about proteins, which do provide us with energy. But carbs and fats are the fuel nutrients in our body. When we eat, we're not just seeking out those three things to get into our body. We also need vitamins and minerals. Vitamins and minerals I don't really talk about in this class too much, but they do play various roles. Okay, as you can see here, vitamins are derivatives for coenzymes, which go into those cofactors. So we need other stuff in our diet besides those three fuel nutrients. Names for enzymes typically are going to end in ACE, unless you have an older enzyme like pepsin, rennin, or trypsin. Okay. So if you see a word that ends in ACE, it's telling you right away that's an enzyme. Okay. We classify them based on what they do, what type of chemical reaction they catalyze, like dehydrogenases, which remove hydrogen, oxidases add oxygen, kinases adding phosphates, ATPase adding or splitting ATP, anhydrases removing water, proteases digesting proteins, lipases, same thing, but for lipids, triglycerides. Some of these we'll talk about a lot in this class, like when we talk later in the semester about digestion. We'll talk a lot about the various lipases and proteases. We'll look at like myosin ATPase, which is important for muscle contraction, found on myosin, one of those proteins we've already talked about. So some of these we get into in great detail. Now, here we can see on the right how enzymes work when they are exposed to what we call substrate. Okay, substrates are the molecules or substances, if you just want to call them a substance, that fit into an area of an enzyme called the active site. Okay, this enzyme is sucrase, which is named nicely because it interacts with sucrose. That's the substrate, sucrose. Fits in nicely into this active site, like a lock and a key, which is the mechanism that they use to describe this relationship. Okay, just like my ring or my key ring, I'm sure you guys have several keys on your key ring. Each key has a specific lock that it fits into. Okay. The shape of the key fits into the specific shape of the lock. The same thing exists with enzymes and their substrates. There's a sucrase enzyme with sucrose. Okay. Maltose and lactose won't fit in here, but maltose will fit in maltase, lactose will fit in lactase. And it is very specific. And what's even nicer, when those substrates fit in there, they actually will have the enzymes will change shape, fitting more snugly around that substrate, kind of hugging it tighter. We call that induced fit. That allows for the chemical reaction to occur, which is what we're showing off here, and ultimately leading to the production of something new, which we would call the new products. At that point, the products are released, and look at our enzyme. It's left just like it was before, ready to catalyze another chemical reaction. All right, 4 things affect and control the activity of enzymes. Remember, enzymes are proteins, so they are sensitive to 2 things. It has everything to do with denaturation. So you have temperature and you have pH. Okay, like I said, all of our proteins have to be sensitive to 1 temperature, which is overall our body temperature. These enzymes listed are no exception. Okay? Enzymes are going to operate best right around where our body temperature is going to be. Should make sense. 37 degrees C is where enzyme activity is almost at its peak, which is what we're showing off with this graph. Rate of reaction basically is telling you how well the enzymes are working. The higher it is, the better off the enzymes are working. We can actually increase body temperature and it will maximize enzyme activity. But if you increase temperature too high, guess what happens? Really steep drop off. Enzymes stop working. Same thing if we lower body temperature, enzyme activity really decreases. What does that mean? Well, as we know, enzymes catalyze chemical reactions, but for all other proteins, they stop working, and we know how important proteins are to our overall body function because they do so many different things, illustrating why body temperature has to be so precisely regulated. We don't want denaturation. Okay. pH is something that's a little bit more variable because different chemicals in our body in different locations will have different pHs. Like our stomach, which has a very low pH. Pepsin is an enzyme we release in our stomach, operates best at that low pH. Anything higher or lower, it's not going to function at. Trypsin is an enzyme that's found in our intestines, small intestines. Okay, it operates at a much higher pH. And if you tried to have trypsin released in the stomach, it wouldn't work. It would be denatured and therefore rendered useless. Other things that affect control of enzyme activity include the concentration of cofactors and coenzymes, concentration of enzyme and substrate molecules, and then there are various stimulatory and inhibitory effects of some products of enzyme action on the activity of the enzymes help to form these molecules. So, kind of long-winded word or sentence that I'm not going to get into too much. But those are all the 4 activity influencing things related to enzymes. Last but not least are the nucleic acids, which are the huge organic molecules containing our CHON, C-H-O-N, carbon, hydrogen, oxygen, nitrogen, but also a little bit of phosphorus, P. And that's in the ATP. Nucleic acids are key molecules for human survival because they provide the building instructions for proteins. And once again, proteins are the things that do everything in our body. Well, we have to make proteins. These guys tell our cells how and tell our cells which proteins. DNA and RNA are the 2 examples in our body, deoxyribonucleic acid and ribonucleic acid. DNA is different from RNA, not only in structure but function. DNA is described as our genetic material inside of our cells. We inherit it from our parents. And just like our inheritance of DNA, we inherit the genes that are located on specific segments of these DNA molecules. Genes are going to control the way that we express our traits. They code for traits we inherit from our parents, and that codes for the synthesis of various proteins. An easy example of this is our skin color. If you have fair-skinned parents, you're going to be fair-skinned. If your parents are dark-skinned, you're probably going to be dark-skinned. It has everything to do with the type and amount of melanin you produce. And melanin is a protein. Okay. Well, you don't inherit the protein from mom and dad. You inherit the instructions for making the protein. And that is the DNA that you get from mom and dad. But DNA doesn't make the protein directly. We use DNA to make RNA. RNA is like DNA's cousin, which is then going to transfer that information to the making of the proteins. So ribonucleic acid is going to relay that instruction from genes to guide each cell's synthesis of proteins from amino acids. Both DNA and RNA are made up of nucleotides, which are the monomer units of nucleic acids. Here on the next page, I can show you a picture of DNA, and I can show you what a nucleotide looks like. Okay? We've got several of them actually up here. We're looking at just a small segment of DNA. Okay. Every nucleotide is going to contain three things: a phosphate, a pentose or 5-carbon sugar. That's actually a monosaccharide in there, so nucleotides are kind of hybrid sugars. And then a nitrogen base or nitrogenous base. Now there are 4 different types of nitrogen bases in DNA: thymine, adenine, cytosine, guanine. There's a different one in RNA called uracil instead of thymine. Not that that's important. But that right there, if I circle this one, would be a nucleotide. Here would be another nucleotide, another one, another one, another one. Okay. And our DNA, as you can see here, is made up of many of those put into 2 strands, because DNA is double-stranded. Okay. And the backbone of the DNA molecule strands are formed from these sugar phosphates. The rings, if you think of like a ladder, or like the rungs, not rings, rungs, are the nitrogen bases. The reason that DNA forms itself like it does and coils up are because of bonds that form between A's and T's and C's and G's. What type of bonds? Anybody guess? Not covalent, not ionic. That leaves us with hydrogen bonds. Not that that's super important, but it's kind of cool to see. If you take biology, you will learn a lot about DNA. Our very last chemical is an organic compound that we've already mentioned in this class for a couple of lectures now, and that's ATP, adenosine triphosphate. It's kind of like, it's really similar to DNA and RNA. It is basically a nucleotide with 2 extra phosphates. It has adenine, just like DNA did. That's a nitrogen-containing base. It has a 5-carbon sugar. The sugar in this case is ribose instead of deoxyribose, like we saw on the previous page. Instead of just having 1 phosphate like DNA and RNA do, it's got 3. That's why they call it triphosphate. Now, each of those phosphates is negatively charged. We've talked a lot about opposites attracting. Like charges repel. Okay. So it's like having 3 kids in the back seat of a car. I experienced this as a kid. There were 4 of us kids. 1 of us had to ride up front. The other 3 got to ride in the back. A big old boat of a vehicle. I was always in the middle, and I always hated it. And big sister and big brother, we all fought the whole way anywhere. And there's a lot of stress placed in the back seat. A lot of stress between these like charges. They even illustrate that with these kind of bended or misshapen lines between the 3 phosphates. This last one here, like the runt, is going to get kicked out sometimes. It gets transferred, instead of kicked out to something else, transfers that energy along with it. That powers anything in our body that requires energy. Things like muscle contractions, chromosome movement during cell division, structures that are moving in the cells, transport across membranes, and even those synthesis reactions that we talked about the other day. When that phosphate's transferred to something else, We then are left with 2 phosphates that would be known as ADP instead of ATP. We'll talk a lot about ATP in this class. Later in the semester, we'll learn more about this, which is cellular respiration, the process where ATP is formed. Where you take an ADP and a phosphate, you attach that phosphate to it, stick little brother in the back seat with his 2 siblings, and you are forming ATP. It's done in 2 phases. The anaerobic phase, which requires no oxygen because there is very little oxygen or none in the cell. Okay. This includes a process called glycolysis you've maybe learned about before. Okay. The word anaerobic is something that we'll see in this class. You can always just associate that as without oxygen. Same thing for aerobic except with oxygen. This is where we produce the most amount of ATP, and this is what takes place in the mitochondria inside our cells. We'll talk about this in Unit 6 after we've talked about the respiratory systems and digestive systems, which provide the ingredients for the making of ATP. So we'll get to that on down the road. Any questions?